how does a T cell recognize a healthy tissue and then attack it, right? We don't want it to do that. We want it to actually be able to turn it off. But it's a fine balance in the immune system because we don't want to turn the immune system all the way off, right? Because then other things will happen like infections and cancer will grow. So what we're trying to do is we're trying to find the subtle differences between T cells that are pro-inflammatory when they're not supposed to be, or T cells that are anti-inflammatory when they're not supposed to be, and look at their signaling pathways and say, is there a way that we can target one or the other and change its behavior in the body?
Welcome to Innovate Law. I'm Antoinette Konski, an intellectual property partner with Foley and Lardner. Join me as we learn from the minds who are shaping the ever-evolving world of the innovation landscape. Today's guest on Foley and Lardner's Innovate Law is Dr. Sam Myers of La Jolla Institute for Immunology. La Jolla Institute for Immunology stands as a pioneering research institution dedicated to unraveling the complexities of the immune system to advance human health. LJI boasts a distinguished legacy of groundbreaking discoveries in immunology, spanning from fundamental insights into immune mechanisms to transformative developments in vaccine design and immunotherapy. With a collaborative spirit driving its diverse team of world-renowned scientists, LJI continues to push the boundaries of immunological research, aiming to translate fundamental discoveries into innovative solutions for a spectrum of diseases, from infectious illnesses to cancer and autoimmune disorders. Dr. Myers is a distinguished researcher at La Jolla Institute for Immunology. With a passion for unraveling the intricate workings of the immune system, Dr. Myers' groundbreaking work has shed light on key mechanisms underlying immune responses, from the identification of novel immune cell subsets to the elucidation of signaling pathways crucial for immune regulation. His innovative research spans diverse areas, including infectious diseases, cancer immunotherapy, and autoimmune disorders. Welcome to our program, Dr. Myers. So glad to have you here.
Thank you very much. Thanks for having me.
So to get started, I think it would be really useful for our audience if you could maybe give us some background and what the overarching theme is that's guiding your research.
Of course. So my work, what I'm really interested in is that I'm interested in the post-translational control of gene expression and cellular decision making. And what that means is that cells in the body are always sensing their environment and their internal environment, and they have to make decisions based on what to do, right? So if an immune cell bumps into a cancer cell, it has to say, oh, I recognize you as a cancer cell, and I have to turn on the genes to respond to that. Or if it's not responding, that's a problem too. So what my lab does is we work on the ways that we can kind of understand how the cell is processing that information and making those decisions. And that information processing, what we call it, is through these biochemical networks. So we study these signaling pathways of these biochemical networks that drive gene expression and underlie cell decision making.
Would you be able to give us a few examples of diseases or conditions that might be impacted by your research? Absolutely.
I will say we are very much a basic research group. So a lot of what we do can be applied to other types of disease research or even basic biology. I'm particularly interested in alleviating autoimmune disorders.
Mm-hmm.
Right? So things that people are suffering with for years and years. So we use a type of analytical chemistry that can look at how a T cell, for example, so T cells drive a lot of autoimmune disorders. Right? How does a T cell recognize a healthy tissue and then attack it? Right? We don't want it to do that. We want it to actually be able to turn it off. But it's a fine balance in the immune system because we don't want to turn the immune system all the way off, right? Because then other things will happen like infections and cancer will grow. So what we're trying to do is we're trying to find the subtle differences between T cells that are pro-inflammatory when they're not supposed to be, or T cells that are anti-inflammatory when they're not supposed to be, and look at their signaling pathways and say, is there a way that we can target one or the other and change its behavior in the body? So one of the things we work on is graft versus host disease.
Mm-hmm.
Right? So if you get a heart transplant or some kind of transplant, you typically have to take a lot of immunosuppressive drugs. How could we do something that would be a more precise target, right? Like how could we stop the graft versus host disease without compromising the rest of the immune system for for cancer or infections or things like that. Another thing that we spent a lot of time thinking about is cancer immunotherapies, which have become really popular in the last decade or so. Again, it's the same kind of thing. We want a T cell to be inflammatory in a tumor microenvironment, but we don't want them to stay inflammatory if the tumor goes away, because then we'll have autoimmune disorders too. And then the flip side of that coin is that we have anti-inflammatory T cells that actually promote tumor growth, right? So how could we stop those from doing what they think is their job and let the pro-inflammatory T cells come in and attack that tumor or that cancer? So those are the kinds of applications that we think about a lot.
Sounds like very important research that could have impact on a wide range of human diseases and really be a benefit to many people. So thank you. Thank you for the work that you're doing. Now, I understand in connection with that work, you developed a high throughput platform to identify the post-translational modifications in cell signaling. Can you tell us a little bit about this platform and why it's so exciting to have something like this to, the science on initiative who don't understand the pace of basic research?
Absolutely. I mean, I can tell you lots about it, but I'll keep it relatively short. So one of the things that my lab can do is we are a mass spectrometry lab. So what that means is it's an analytical chemistry tool where we can look at little chemical modifications that get added to proteins, and those chemical modifications affect their function, right? So that turns it on or off, or causes it to be degraded, or causes it to be more stable, right? And typically, immunology labs and cancer biology labs, they'll study a handful to a dozen of these post-translational modifications. And then they'll describe the cell and how it's behaving based on a dozen landmark, what are called phosphorylation events, or small phosphates that get added to proteins. In reality, it's 20 to 30,000 phosphorylations that are changing or that are occurring in the cell, and tens of thousands of those can be dynamically regulated, right? And so with mass spectrometry, you can measure 20,000 phosphorylation sites, but the problem is that each one that we want to study is basically an entire graduate student career. I studied one in graduate school, and it took me 6 years to figure out what one of them was doing. And that was a big risk too, because was it even important in our system? So the technology we've developed recently is called, it's post-translational modification-centric base editor screens. And basically what that means is that we take the one phosphorylation site that might take a couple years to decide if it's even important. We can now look at 10 to 20,000 of those in a single experiment and rank them based on their importance, right? So we have this unparalleled ability to discover important phosphorylation events that before were either, too risky to try something new, or there aren't any good reagents for it, so we never look at it. And because of that, we end up looking at the same things over and over again and explaining these incredibly complicated systems through a handful of landmark phosphorylation sites. So with base editing now and these PTMs-centric base editor screens, again, we can functionally characterize tens of thousands of phosphorylation sites in a single experiment. And that allows us to go in and figure out, what are the new signaling events or biochemical networks that we weren't looking at before. Now we know these are important and we need to go in and study these more. So
that's remarkable. Why hasn't this been done before?
I don't know. I think part of it is, as science is becoming more interdisciplinary, where you're not just good at your one thing anymore, you're kind of learning a lot more. But what that ends up looking like in real life is that, one lab knows how to do one thing and another lab knows how to do a different thing. I've always kind of been really excited about bridging the gap between disciplines, or like being a liaison. And so in my postdoc, so I've always trained in these mass spectrometry labs. I've been in mass spectrometry labs for almost 20 years now. But in my last position, I was in a genomics institute, right? And so they approach things very differently. And so when David Liu invented these base editors, I was in that institute when those came out. And because I was unsatisfied with the fact that we can measure 20,000 phosphorylation sites, but I can only study 1 per decade or 2 per decade, right? It really became a thing that I was able to connect those dots. And because I liked learning more and more about, either parallel, even not parallel disciplines, I was able to piece those things together. Now, why no one else has done it before, I maybe that's part of it. But yeah, I think I was just in the right place at the right time to have this light bulb turn on. So
That's good. I mean, obviously it was your vision and your ability to synthesize these different, very at the point discrete disciplines and bring something together. But I'm sure LJI's collaborative approach also is very useful to the work that you do. So,
it's been totally amazing. I mean, I did a lot of the kind of foundational work at the Broad Institute, but most of the project we got done here with help from the sequencing core here and the flow cytometry core. Like we've just had incredible collaborations with people all around. So
that's great. So, from a basic science point of view, that's very interesting. But, how does this translate for wider applications? Do you find new druggable targets? Do you find new pathways? Is it used for personalized medicine? What's the applications?
I honestly think the applications of it are going to be very widely applicable. It's not a great line. I actually think the applications are going to be very widespread. So, it can be used in cancer biology to understand which signaling events are necessary for the cancer cell to grow or proliferate, right? If we can make mutations of those and see them die faster than the rest of the cells, that might be a target that we can look at. And then the computational and bioinformatic tools to understand signaling are getting better and better. They've been great for genomics for decades, but they're really only becoming usable now in mass spectrometry and proteomics. So we're taking that kind of fledgling bioinformatic knowledge that's coming out from mass spectrometry finally, and we're actually porting it back over to genomics. And so that would allow us to identify potentially signaling pathways or kinases that we could inhibit to alter a cancer cell growth. And the reason I think that is so powerful is that, most of the drugs we have right now are against signaling molecules, right? So if we can look at signaling at a resolution commensurate with its actual complexity, we can start to actually, not just take a sledgehammer and, hope we're hitting the right thing, but we can actually really go in with more precision and say, well, I need to turn off this pathway a little bit and this pathway a little bit, and that'll give me the effect that I'm looking for. So it'll be helpful for cancer biology. I think we're already making it useful for immunology. I could see it working in neurodevelopment disorders. We've done some work with Tony Koleski at Yale, and we can see signaling pathways that are disrupted in developmental disorders. And these would be things that we could now, better understand the pathway and go in and try to use existing drugs and repurpose them for new things. So I really think it's going to have an effect on all basic biology and biotechnology. So.
Well, that's really interesting, how you were using it to apply that in drug discovery and development and finding new, maybe even repurposing existing drugs for new disease applications. And you've done so much already with your career. What's the future direction of your research?
So we're really excited about this base editor screening technology because it allows us to close this loop, the scientific loop where, you make an observation, you make a measurement, you model your measurement, and then you have to go and predict it, right? We haven't been able to we haven't been able to perturb it at the scale in which we need to check our guesses, check our hypotheses, right? And so with mass spectrometry being what it is now, we can look at 20,000 phosphorylation sites, and we can use RNA-seq to look at all the genes. There are computational models that we can meld those, and now we finally have the technology to make the perturbation and say, is my prediction about the way this network is integrated, is that correct? And if it's not correct, that's fine. You know, we go and we try something new, but now at least we have a technology to do that. So what we basically want to do is apply to everything we can get our hands on. So my lab does a lot with T cells. I have a collaborator at University of Pittsburgh who does a lot with macrophages. They're kind of like an innate immune cell that's one of the first responders in infection. But again, we can apply this to a lot of things. So what I'm hoping to do is make this some kind of platform where anybody who was like, well, how does my cell decide to go this way and not that way, right? We can then set the experiments up and set this whole model up to say, well, let's measure it going that way and then see which ones are actually important for it. So really, we just want to apply this to as many things as possible.
That's wonderful. So if any member of our audience wants to learn more about your work at LJI, how can they do so? Where should they go?
So there's two main things. So one is the LJI website itself has a lab page for everybody on there, and I'm on there as well. And we try to keep that updated with, you know, recent publications and group members. And then I've created another website just for the lab that's kind of independent of LJI. And what we're trying to do there is add a lot more data and information people can use. So, you know, if you need a protocol, if you need a publication, you don't have access to it, you can go to our website. It's SAMYERS lab, so samyerslab.org. And you can go there and you can get our papers, you can get some protocols, you can read about what we do. And then our contact information is on there as well. So if you do want to reach out, you're more than welcome to.
Thank you so much for all that you're doing and the research that you've undertaken so far. And we really look forward to following your career and your future discoveries. Just thank you.
Thank you. Thanks for having me. It was a pleasure talking to you.
Thank you for watching Innovate Law. We hope you found our discussions enlightening and inspiring. Join us next time as we continue to explore the latest trends and share compelling stories on the cutting edge of innovation.